This demonstration uses 2 and 3D live cell imaging to understand the spatiotemporal relationships of proteins involved in the DNA damage response. First, transfect or transduce your cell line of choice with expression constructs containing appropriate fluorescent markers, here M-cherry and GFP. Next, acquire a series of control videos showing the fluorescently labeled cells under normal conditions. Then apply an appropriate treatment and capture data of the treatment effects on the fluorescent cells. Process and analyze the video data for spatiotemporal relationships of proteins. Ultimately, live cell fluorescence microscopy can detail cellular dynamics, like the formation of 53BP1 foci in response to DNA-damaging agents, or 53BP1's behavior during mitosis.
We first became interested in 2 and 3D live cell imaging when we wanted to study how varying treatments affected mitosis in certain cell lines. This experimental approach allows us to study the DNA damage response and the maintenance of genomic stability more easily than traditional methods. Additionally, you can use this method to study how certain proteins interact in various signaling pathways in various cell lines. The main advantage of live cell imaging over traditional techniques like immunocytochemistry is that it allows the study of the DNA damage response proteins in real time. Optimizing the right recording conditions can be challenging. This demonstration can be used to see the many small steps needed to ensure success. Remember that persistence pays off when it comes to minimizing